Started. So, hi, welcome everyone. So, today we have Tabish with us, who is going to talk about his PhD work on cellular geometry of the heart. So, that will be a lot of shape analysis. Tabish is a postdoctoral fellow at the Center for Research in Neuroscience at McGill University Health Center. Uh, he also did his PhD at McGill in the Department of Computer Science, where he worked with Kaleem Siddiqui on the reconstruction of biological tissue geometry from 3D microscopic images. And his research interests include applied mathematical methods for computer vision, image reconstruction, and image representation. Tavish, you can take it away.
Okay, thank you very much, Pulkit, for inviting me and letting me share my work. So, if you guys haven't looked at the paper, I'm going to talk about two papers. They're from my PhD work, as Pulkit said. And the general topic of the talk is cellular geometry of the heart. And I'll talk about the mammalian heart, specifically mice heart. This is slightly different from maybe the kind of work you would see in McCai uh, type conferences, and uh, and this is based on like two papers. One is our work in 2019 in ITME, and the other one is in what is called the EMBO journal. Perhaps people on maybe people are not so familiar with this because it's a molecular biology journal. Uh, so I wanted to talk about this for a few reasons. One, geometry of heart is interesting, and second, because a lot of people in computer vision and medical imaging, etc., are unlikely to read this journal. It's a very biology journal, and then for a computational person, even for me, it's like hard to parse what's going on. So I'll basically go into the computer vision parts or the medical imaging parts of the work, which are not as apparent if you read the um, actual article and the EMBO. Okay, so let me. So before I get started, as Pulkit said, if you have questions, please post them in messages. I'll try to answer, and stop me whenever. And also, like in the end, if you have questions, like you can ask me then. All right. So, uh, so I'll first introduce basically what models of the heart exist, then talk about how they arrive at those models, how our way is different and also similar to what they did. Then I'll talk about doing this in presence of noisy images, and then finally talk about organization of heart tissue at the tissue scale, and then some future work and ongoing work that we have in this field. Okay, uh, so heart uh, is an interesting organ to study. There's a lot of diseases. There's a lot of it's an extremely important organ for functioning of human body and also in animals. So there's a lot of different models that have been proposed, and and from like very early on, like in like different people have come up with different models. Earlier models typically used to be based on anatomical sections. So people would cut open heart, look at them, and then come up with some models. Uh, so here's like a few. So for example, this one is a quite early one, which looked at heart, excised them, and then like dissected them, and then came up with like this three-layered model. It's all the way from 53. So they didn't have fancy equipment, they didn't have fancy machines or microscopes to actually look at it. They just excised it, looked at it, what they had, and then came up with this model. But there are others which are kind of popular. So, for example, this model uh, called the ventricular band model, they propose that heart basically is like a single sheet, and then it wraps around itself to form like a four-chambered thing, and it pumps blood. it's kind of attractive, and like if you look at these pictures and then like twisting and stuff like that, you would think that, oh, this sounds like really nice, but it doesn't make too much sense from from developmental point of view. uh, there are other models which again, like people have cut up tissues, looked at them under a microscope, or looked at sections of them, and uh, this is, I would say, roughly the current model that people go with, which is the cleavage plane model. That is, the heart cells or the muscles of heart are arranged along cleavage planes, or along planes or sheets, and they are stacked on top of each other. Uh, so all these models are based on some form of uh, looking at the heart tissue with maybe some magnification or some sort of naked eye and stuff like that. However, like there's this like fancy imaging you might have heard of called diffusion tensor imaging, which can be used to infer um, the structure of the heart without actually looking at it. So diffusion tensor imaging, if you don't know, what it does is it measures the speed of diffusion of water. So if you have something that is uniform, something which is like on the top right, water will diffuse uniformly throughout this. If you have something that is not uniform or is anisotropic, say something at the bottom, there's some anisotropy, water will diffuse non-uniformly. So for example, in the first one, water can go anywhere. In the second one, the water has a preferential direction. Now, because heart cells are kind of cylindrical, they have a preferential direction of diffusion of water, and then you can use it to infer the orientation and the structure of how muscle fibers are organized. And people have used this to measure like uh, this diffusion tensor everywhere in the heart. It's typically used in the brain where you have fiber bundles going from one end to the other, but you can also use it in the heart, which has muscle fibers. Uh, so 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 this measurement is kind of it's not a direct measurement. It's like an indirect measurement. So we use the direction of diffusion of water as a proxy for direction of the cells, and assume basically that the direction of cells is the direction of diffusion of water. And also, this diffusion is done at like a very coarse scale. So you would have like this entire block, for example, this size, well, actually kind of this size, and you would measure the average direction of diffusion. However, if there is some multiple directions and stuff like that at like microscopic scale, you can't really pick that up. Uh, so typically, because the cardiomyocytes are kind of cylindrical, you would expect the diffusion tensor to be sort of ellipsoidal. So you have more diffusion along one direction and kind of similar uh, level of diffusion in the other two directions. And you can use this information to kind of estimate the orientation of cells at different regions. You take this orientation at different points and then put them together. What was kind of popular, what is called myofibers. So because these cells kind of stack one after the other, uh, so you can talk about these myofibers, or fibers along which this water diffuses. Now, going back to the cleavage plane idea, so people believe that cells, these fibers, kind of arranged along sheets, and these sheets have cleavage planes in between them. However, this is again like this is very qualitative, so people basically look at this. There's no quantitative way to actually measure this sheet, or there wasn't any quantitative way to measure this sheet-like organization of fibers. Uh, so to summarize, so what people do to get at the structure of the heart is they'll measure diffusion. They use diffusion tensor imaging to measure average diffusion of water in different regions of the heart. Now, this diffusion is directional, so you have more diffusion along one direction, less in other two directions, and they pick the direction where there's most diffusion as the direction of the cell, and then assign orientation of cells in different regions of the heart. Now, this is done at a millimeter scale, which is like hundreds and hundreds of cells in a single block. What we wanted to do was, instead of using this diffusion and like these fancy DTI machines, we thought, okay, we'll just do something very simple, which is we basically just look at the cells themselves and use microscope to just look at them and then like estimate the actual orientation of the cells. And that is what we did. So we section the heart, we take like an entire section, and then use confocal microscopy to image the tissue in 3D. So you have a 3D image of the entire tissue of all the cells. Once we have that, what we can do is, so if you look at the cells, you'll see that the intensity of the images kind of doesn't change much along the cells, but it changes very rapidly across the cells. So using this simple idea, we can estimate the direction or the orientation of each cell. So least change is the direction of the cell, least change in intensity is the direction of the cell, and the most change in intensity is the orthogonal direction. So once you have this, you have direction at each voxel basically, and you have this at the cellular scale. And this is the actual true orientation, and then you can get it everywhere, wherever you have your imaging data. We do this for entire section. So we take the full heart, we clear the tissue, which is we make it transparent. So now we can image the whole tissue. We can image whole tissue in 3D. Then we section it into like thick sections, put it under a microscope, and then image the whole plane. Okay. This process is kind of non-trivial. I'm not going to go into this. This is there's like a lot of biological detail here. It took like 2 or 3 years to actually define this process. But from a computer vision point of view, what we have is 3D images of like an entire section, and we need to stitch them together. Basically, there's some overlap, and we want to make sure that they. So this is like a traditional registration problem. It's traditional registration, except with some caveats, which is the structure is kind of very repetitive. So if you look at the boundaries, there is no clear markers like you see in various different registration problems. So they'll have clear points of interest or some anatomical landmarks which you can use to register. However, here you have repeated patterns of cells which look very similar everywhere. So in order to register this and to register this cleanly, we used phase correlation because the pattern is very much like a cosine sequence or a sinusoidal sequence. And you can use phase quite effectively to actually align such images. And it works like really, really well. And it's hard to tell from this image where the boundary of the two tiles is. So you can, like, it's very difficult to see any seams between the images. So using phase correlation, we are able to stitch them like really well. Okay, so once we have this stitched over the entire section, in the ideal scenario, we would have like nice block of tissue all throughout, because we had like transparent. Like in theory, we had transparent tissue, so we could like image it throughout. But as it turns out, it's not really transparent. It's kind of it's not like completely transparent. It's slightly yellowish, as you can see. So what happens is when you image this, you get really nice images up top, but not so good images deep inside the tissue. So this is the classical noisy problem that you have in various different scenarios in medical imaging. It's slightly different here because you have a block of tissue which have clean images towards the top and really noisy images towards the bottom. It's the same structure with variable level of noise inside the image. So how do we fix this? So on the left, you see images towards the top, which is clean, relatively clean, and then on the right are images which are noisy and are deep inside the tissue, and it's difficult to estimate the cellular structure there. Uh, so here is the distribution of noisy images, and this is what the distribution of clean images looks like. And if you look at this kind of gave us the idea of using like transfer learning kind of approaches where we want to change one distribution into another distribution. So we came up with a pipeline based on CycleGAN. It was popular at the time. So the idea here is that, so there is no ground truth, but what we know is that what we're imaging is the same tissue. And we know that tissue is clean towards the top and then noisy in the bottom. So we want the distribution of images in the bottom half to look like distribution of images in the top half. So it's like an unpaired image-to-image translation. Clean images in the top, noisy images at the bottom, and we want to change clean images of top to look like noisy images at the bottom to look like images that are clean. And we pose this as like a simple image-to-image translation where one domain is noisy images, another domain is clean images, and we want to transfer one to the other. Okay, we do that. We use clean set to train, and then noisy set. Noisy set is like we want to like change noisy set into clean images. And here is some examples. So it works pretty well. For example, on the left image, you see that it's kind of blurred out in the middle, but you see structure image at the bottom. So you start to see cellular cell boundaries. Again, also on the second image. So things that were washed out are kind of now more clean. However, one problem that we saw was that if the images were really noisy, or even sometimes in non-noisy cases, there were many hallucinated edges. So for example, in the right image there, you have edges that go across as well as along the cells. Now, this was not good for us because we wanted to infer the structure of the cells. But if our method kind of introduced these fake edges, then our estimation would not be reliable, or it won't be trustworthy. So instead, we decided to basically just dump this and use a more simpler and a more traditional approach of dictionary learning. So we take the clean set and learn a dictionary of patches on the clean set, and then use this learned set of patches to clean the bottom images. Now the results, looking at them, are not that great, or not visually that great, but you don't see any hallucinated edges. So the noise is gone, but you have like a slightly washed out look, but you don't. But the good thing is that you don't actually create any fake edges, which would be really bad because we don't want to have like false structure. So we went with this method. It's a method that's like more trustworthy, and use this to estimate the orientation using what is called the structure tensor. Now, structure tensor is like similar in theory or in principle to what DTI kind of does. So DTI measures diffusion of water. Structure tensor is gradient of it's constructed from gradient of the intensity. So direction in which gradient is the least will have smallest eigenvector, and the direction in which gradient is the largest will have large eigenvectors. And we want to pick the direction in which gradient is the smallest, which is along the cell. So we use that and estimate the orientation in like a tissue log. Okay, so let me recap. So we wanted to construct the cellular geometry of the heart at like by using microscopy, so that we get true orientations and not some proxy. So we took a tissue, cleared it, just make it transparent, put it under a microscope and imaged it using, so because it's like a huge section, so we image it in tiles, register those tiles. Then we have noisy images in the bottom, clean kind of on the top. So we want to denoise them. So we use a dictionary-based approach, which kind of gives us denoised, but also no fake edges image. Use that to estimate the direction of cells in this 3D block of tissue. And then we have cell orientations at every point. Okay, so we use this to, we do this in short axis, long axis, also in apex. So traditionally, a lot of the models have only looked at ventricles and also like very small parts of ventricles. As far as I know, I think we are the first ones to actually look at apex and also look at cells at such like almost like 100 times more resolution than is typically done in other models. And also we look at the entire section, not just like a small region. So we can basically look at, so now we have the entire picture. So we have what happens in the cross section, what happens in the long section, and then what happens at the apex, which is kind of has been mysterious of what's going on there. Okay, so there's some interesting results that come out of it. So so one of the ways in which people have measured the structure of the heart, or the geometry of the heart, is what's called the helix angle. So people talk about how the cells are oriented, or how the orientation of the cell changes when you move from the epicardium, which is the outer wall, to the endocardium, which is the inner wall. When you look at it at the cellular scale, you see that, so you see this is what the orientation kind of looks like. So from outside on the left, inside on the right. If you do this using DTI, you get something like that, which is like a smooth line from left to right. But if you actually look at it at the microscopic scale, you see that, for example, in the first part, you have, let's say, a flat region, then really rapidly changing, and then again a flat region, and then it changes again. So the rate at which the cellular orientation changes is not uniform. But if you did this at a coarse scale, maybe at like a DTI scale, at like sub-millimeter scale, what you would get because of averaging across big regions is a smooth variation, or like a straight, a constant change from outside to inside. So we gain some like newer insights on how the cells are organized at the microscopic scale. Okay, so now that we know about the orientation of like individual cells, we can talk about this idea of sheets and this cleavage planes. So let me recap what the idea was. So there's this theory that cardiomyocytes organize in fibers, that is, they're end to end, and then form kind of fibers, and then these fibers are organized along sheets. Okay, and then the sheets are separated by what are called what is called in the literature as cleavage planes. Now this has been very qualitative, so people haven't really measured this or like shown it in any quantitative way. So we wanted to kind of like quantify this, like, do these sheets exist, and like how do you actually talk about them? And that is what we kind of did. So let's formulate this problem as a mathematical. Yeah.
There are a couple of comments in the Q&A if you want to read them. And
Oh, okay. Yeah.
yeah.
I don't see the messages, so yeah. So
Oh,
please.
you cannot see the Q&A.
Yeah,
Okay,
I can.
no worries. I think one question is, what do you mean by bottom images and the top images, and which axis? And could you also comment upon what is the cause of different signal to noise ratio for these two images, these two types of images?
Okay. So let me. I think it's referring to these ones. Okay, so let me go all the way to head. So the imaging is done using a confocal microscope. Now, confocal microscope is a light microscope, which is it uses light to image, right? So you stain the tissue. It has a fluorescent molecule. You shine some light, and then it reflects, and then you capture that light. It's a 3D imaging, 3D microscopy technique, which is like you actually image like an entire block of tissue at once. And the way it works is you change the focal plane of the image of the image that you're capturing. So you have a 3D block, and you change the focal plane of your light to image in 3D. Okay. Now, if your tissue, so you can only do this when your tissue is transparent, right? Because light has to go through and then you reflect back. So if your tissue is not entirely transparent, then what happens is light has to go through this translucent tissue and is modified or like gets scattered by the tissue and by the impurities in the tissue, changes staining, etc. So it goes all the way down and then gets reflected back and then also has scattering and all other stuff. So what happens is like towards the top or like closer to the lens, you get clean images because light doesn't have to go through this translucent medium or translucent tissue. But towards the bottom, which is like far from the objective of the microscope, the light, because it has to pass through the tissue, it gets scattered and it's like more noisy. So what you get is noisy images towards the bottom and then clean images towards the top of a tissue block. Okay, and that is what I show here. So you have on the right, on the left, you have images from the top, which is closer to your objective, and then towards the bottom, and then on the right is images from the bottom of the tissue, which is further away from the objective lens. So because it's further away, there's more noise, and because top images are kind of close, there's less noise. And it's difficult to estimate like cellular orientation from like deeper tissue block. I don't know if that was that the question, or did I go on a rant about confocal microscopy?
I think they got it. Yeah.
Yeah. Uh, okay, so that's the idea of like why there is like inhomogeneity in images, and they vary with the depth of the tissue.
And these are all 2D slices, right? Which
No, this is
so
3D blocks. I show a 2D slice, but the images are in 3D.
so the cycle GAN, is it implemented in 3D or 2D slices?
The cycle GAN is in 2D.
Okay. So then you
So
stack them up, uh, the corrected denoise.
yes.
Got it.
Um, okay. Um, okay. So we have orientation of cells for a 3D block of tissue. Now we want to look at how do these single, say, fibers or like single cell orientations organize, or do they organize in sheets at all? And then how do you even ask this question? So as I said, like traditionally, people look at it qualitatively, see, oh, it looks like they are in sheets. So they call them cleavage planes. Um, so way we formulate this question is as follows. So we have the orientation of cells at each point in space. So you can think of it as a vector field in space. So let's say you have a vector field like this, which is circular or cylindrical in 3D. So you are given a single vector field. You want to estimate another vector field, which is orthogonal to this vector field, so that it spans a sheet locally, right? So if you had something like this, which is a circular vector field, which is your given vector field, you want to estimate another vector field which is orthonormal to this, which would be, for example, in this case, axial vector field going along from like along the z-axis, and then the sheets would be ones that are locally tangent to this frame. And then you can do this everywhere, and then you would ask, okay, there's sheets or there's no sheets. Uh, okay, so we get vector field from our measurements from our cells, and we want to estimate the second vector field, which would be orthogonal to these fibers, and then fit a sheet to say whether you can actually fit a sheet or not fit a sheet, or how likely it is that these exist. Okay, so the way we do this is using what is called the Lie bracket. So Lie bracket of vector fields is this simple idea that if you have, let's say, two vector fields, v and w. So there's one w which is going one way, and then v, let's say, there's another way. And then if you form a closed loop, or let's say a parallelogram, so you go in the direction of w, then in the direction of v, then in the direction of minus w, and then in the direction of minus v, you would end up in a different place from where you started. Right? So if you look at the picture on the right, so if you start at P and then move along a loop or move along like two different directions, you would end up in two different points because the fields kind of change in space. And the difference between the two is a vector, and that vector is called the Lie bracket of the vector fields. Okay. Uh, and we use this idea to define a sheet, which is, let's say we're given a vector field u. We want to estimate another vector field v, so that if you make this loop, if you move in this like parallelogram, you end up at the same spot. So basically, parallelograms kind of close. So this is a case where like Lie bracket would kind of be zero because you end up at the same place. This would be a case where Lie bracket is non-zero because you end up in a different place when you move in this parallelogram or you move in a loop. And we look at the distance between the starting and the end point. We look at the norm of that and try to minimize that. Okay, so here's the formulation. So we are given u, which is red. We estimate another vector field v, which is orthonormal to u, and which minimizes the normal component of the Lie bracket. So they locally will form a sheet. Let's say the locally will, the parallelograms will kind of close. So we use like simple gradient descent to do this. It's well, okay. So it's not completely straightforward. There's like subtleties here, but I'll not go into those details. So, but uh, it's just like in principle, it's just basically gradient descent on v. So we basically estimate v. So we have this energy, which is like square of this Lie bracket norm. We find its derivative and then use the derivative in simple gradient descent to estimate estimate the second vector field. Okay, we do this.
Here's a question. Sorry to interrupt.
Yeah,
So could you go back to your previous slide
this one.
where you show the energy minimization term? Yeah,
This one. Mhm.
yeah. So you take the mean squared error argument between the Lie bracket and the cross product. The question is,
Mhm.
could you explain why do we need a cross product there?
So cross product of u and v would be vector which is normal to u and v.
Right.
So you take the Lie bracket between the two, which is a vector, and then you take the dot product with the normal. So you basically project the Lie bracket onto the normal plane. So it's the normal component of the Lie bracket that we're minimizing here.
Got it. Yeah, it's clear. Thanks.
Okay. Um, so we're given one vector field, which is, let's say, red here, and then we estimate the second one by minimizing this energy using gradient descent, and then we get the second vector field, which is orthonormal to this. So once we have that, uh, you can use this local frame to fit like a small sheet at a point. So in this cylindrical case that I started with, like circular vector field, you start with that one, and then you estimate the second one so that it minimizes this energy, and then you're able to find this axial vector field, and then locally fit a sheet which would be tangent to these two frame this frame field. We do this on so this is on DTI data. So we do this DTI data on canine hearts. So we have direction from DTI. We treat that as first vector field, then use our method to estimate the second vector field, and then fit these local sheets on in different regions. And in this picture, I show the sheetlets, and also I show the residual energy. So energy is small in blue regions and high in red regions. So there are some regions where you can fit a sheet, and there is some regions where you cannot reliably fit a sheet. So for example, there's regions on the right which are kind of red, which are places, and also towards the inside, which are the papillary muscles. So they don't support sheet-like geometries. Stuff on the right, which is the right ventricle, so right ventricle, and also near septum, that doesn't support sheet-like geometries. So now at each place, we can like say, does heart cells kind of support this sheet-like geometry? And if they do, we can actually get the second vector field and fit these local sheets on those at those places. We do this for the entire heart. So this was from DTI for the entire heart, and we can do it at every place, and then we can talk about what sheets look like in different regions. Okay, I think I'll wrap up. So before I move to the future, future and ongoing work section, so let me summarize. So geometry of heart is typically done by DTI at coarse resolution, or by looking at anatomical sections, or like single planes, or like visual inspection and qualitative descriptions of the heart. What we wanted to do was do this at the cellular scale. So we actually wanted to look at each cell and then measure the orientation of each cell, and then use that as the direction of the cell. Do this the entire section, then look at how that is organized, then look at the prevalent sheet theory, or the cleavage plane theory of the heart, and then try to quantify it. Like first formulate it as a quantitative question, and then like estimate regions which support sheet-like geometry and regions that don't support sheet-like geometry. Okay, so there's like a lot of questions still unresolved. There's this. So the obvious one is that heart is not a static organ; it moves, it beats. That's the whole point of heart. And the actual orientation of the cells changes in beat cycle. So this is something that we would like to do. We have like some preliminary data on this, but not so much. Where the actual cell orientation changes in beat cycle, and that is how cell kind of like contracts and like is able to pump blood. So we would like to do this in like a normal beating heart to actually track how they change in time, and then what happens to sheets in time, and what happens to cellular orientation in time. Another thing that we noticed during our imaging, so we have some hearts which are like diseased, they have like specific mutations. The geometry actually changes, and the changes in geometry is different in different diseases. And it's in the actual orientation of the cells. So we can characterize how much different diseases deviate from like regular geometry. So this is again like future work. We have like some like we had like some diseased hearts, like we had some like preliminary qualitative data on like it looks different, but we haven't actually quantified this, and we'd like to do that. Finally, we also would like to like see where this orientation comes about and look at embryonic development. This is something that is like ongoing, and we had like a recent paper on this like last week, where we look at parts at different embryonic stages, and then use that to measure the orientation at different time points, and then see how this this geometry kind of emerges in development. This work was still like on a small piece of the heart, not entire heart, but that is the plan is to actually extend this and then do this on the entire heart and then see how this geometry kind of arises. Okay, and with that, I think I'll wrap up and then take your if you have any more questions. Are there any questions?
Doesn't look like it. I think you answered all the questions that were in the chat.
Oh, okay. So because we had this video, I'm going to play it. And yeah, that's geometry of the heart at the cellular scale. Uh,
Yeah, that look,
so this I'll end at this frame and like it's like really striking. I kind of like this video because it's like really striking to look at how the cellular structure of the heart is at this scale. I had not seen this. I don't think anyone has, but yeah, it's cool. But yeah, I hope the audience liked it. Yeah.
yeah, no, that's very cool. Do you know what other work, what similar work is being done on some other tissues in the human body? If not heart, some other orientation work.
Um, I
Cellular level.
don't know. So I don't know of orientation work per se, but like specifically embryonic development, there is a lot of people like who are interested in like seeing how this kind of happens because a lot of heart diseases are like happen. A lot of congenital heart diseases happen because the geometry gets disturbed at some point, and there's like a lot of interest in understanding this mechanism, and a lot of work on embryology is typically very early stages of embryonic development. What we are looking at, or like what we are interested in, is like later stages of where this like geometry comes about, and then like see how like the kind of questions that we are interested in is like the optimality of this structure and how this uh, the specific geometry of the cells is optimal in pumping blood or in on mechanical strength and stuff like that. There's like similar work in like very different contexts. Like you see stuff like this in, for example, a bone like organization of bone, uh, uh, organization of crystals, for example. So there's like stuff like this. So it comes up here and there, but the thing is like it's a very it's not like there's no like a central theme, a central like a single conference that talks about stuff that is related to this. Which is also why I wanted to talk about this work, because I know that people won't be reading the molecular biology journal who are interested in like geometry or who are interested in medical images. So yeah.
Oh, yeah, that's very cool. Yeah, it's different than what we usually cover here, so that's good. You always see non-deep learning stuff.
Yeah, I try to throw in some deep learning.
Yeah. Well,
not much deep learning.
well, I think yeah, you answered all the questions, and the recording should be available on YouTube soon. Um
Okay.
yeah, I think with that we should
Awesome.
take care. And thanks everyone for attending on a Saturday. Cool. Well, thanks a lot, Tabish, for volunteering.
Thank you, Pulkit, for inviting me.
Thanks. Bye. Have a good day.
Okay.
Bye bye.
Good day. Bye.